Okay, let's really get into this. 7%, think about that. 7% of our genes are blueprints for membrane transporters and their pals.
It's a huge number when you really stop and think about it.
Right. A huge chunk of our genetic code just dedicated to these cellular gatekeepers. They control, well, pretty much everything that goes in and out, keeping us alive.
Absolutely fundamental.
So today, that's our obsession. We want to dissect these critical proteins until, we have a rock-solid professional level grasp of their role in how our bodies handle medications.
And we've got a lot of great source material to draw on for this.
We definitely do. We've got a serious stack of information to unpack, so let's get started.
It's truly remarkable how central these transporters are. They're not just about basic cell function. As our sources detail, they are absolutely pivotal in determining how we respond to drugs.
Yeah.
Imagine every single one of your cells has these intricate security systems deciding what gets across the border.
Exactly. So we're diving deep into membrane transport proteins and their massive impact on how our bodies manage drugs. We're talking the full journey: absorption, distribution, metabolism, excretion, the whole ADME picture.
And even how drugs target diseases in the first place. That's crucial too.
Right. We'll explore the core mechanisms, get to know the major transporter families, the big ones, ABC and SLC, and understand their roles in both the good stuff,
the therapeutic effects,
and the bad stuff, the side effects. Plus, we'll tackle the fascinating world of genetic variations in these transporters and what those variations mean for individuals.
And if we step back, understanding these molecular machines at this level is really key to unlocking some of the biggest mysteries in pharmacology. Why does a drug work wonders for one person and barely touch another? Why do some people experience nasty side effects while others, well, they just breeze through treatment?
Yeah, the big questions.
This deep dive into transporters is about getting to the fundamental reasons behind these individual responses and, ultimately, paving the way for more personalized medicine.
So where are we headed in this deep dive? What's the roadmap? First, we're going to build our foundation with the fundamental principles of membrane transport.
The basics.
Exactly. What's the difference between a transporter and a channel? What's passive versus active transport? Got to get that clear.
Foundational stuff.
Once we've got that solid, we'll zoom in on the structure and function of the big two, the ABC and SLC superfamilies.
Yeah, the heavy hitters.
We'll highlight some of the headline acts like PGP, CFTR, CERT, and DATE to really anchor these concepts, make them concrete.
Good examples. Then we'll connect these molecular mechanisms to the bigger picture of pharmacokinetics, what the body does to the drug, how it moves through us.
Right.
And pharmacodynamics, what the drug actually does to the body, its effects.
And we'll also address some major challenges where transporters are front and center, like drug resistance in infections and cancers.
A huge...
And those unwelcome adverse drug reactions.
Definitely need to cover those.
And because we want to understand this inside and out, we'll tackle the significant impact of genetic variations in these transporters. These subtle differences in our genes, well, they can lead to really dramatic variations in how we respond to the exact same medication.
It's incredible the difference genetics can make.
We'll also touch on how this knowledge is reshaping drug development and even influencing the regulatory landscape.
Yeah, the FDA guidance and all that.
And we can't forget about specialized barriers like the blood-brain barrier and the unique role transporters play there.
Absolutely critical for CNS drugs.
Finally, we'll explore the extended clearance concept and PBPK modeling. These are really powerful tools that help us understand the integrated function of these transporters within our complex biological systems.
Putting it all together, ready to really dig in.
Let's do it.
Absolutely. Let's start by dissecting how these transporters actually perform their crucial work. A fundamental distinction, as you mentioned, is between transporters and channels.
Right. They're not the same thing.
Not at all. While both facilitate the movement of substances across cell membranes, they operate with completely different mechanisms, as our sources clearly lay out. Ion channels essentially form a gated pore.
Like a tunnel.
Kind of, yeah, like a door that opens and closes. When it's open, specific ions can just flood through, down their electrochemical gradient, at incredible speeds. We're talking maybe 10 to 10 ions per second. It's fast.
Wow.
And these gates are controlled either by changes in the electrical voltage across the membrane, those are voltage-gated channels, or by the binding of a signaling molecule, a ligand-gated channels.
Okay, so channels are the express lanes for ions, open the floodgates, and they zoom through based on charge and concentration differences. Very quick.
Exactly. But transporters are much more, let's say, deliberate.
Okay.
They physically bind to the specific molecule they're moving, forming a temporary complex. This binding triggers a change in the transporter shape, a conformational change.
Right, it shifts.
Precisely. And that carries the molecule across the membrane and releases it on the other side. Now, this is a much slower process. Turnover rates may be in the range of 10 to 10 millimolecules per second, much slower than channels.
Big difference.
And because they have this binding step, transporters have characteristics that channels don't really exhibit. They can become saturated.
Meaning they can get overwhelmed.
Yeah, if there's too much of their target molecule, they can only work so fast, max capacity.
Yeah.
And their activity can be inhibited by other molecules that compete for that binding spot.
Like rush hour traffic versus an open highway.
That's a pretty good analogy, actually. And the driving force behind this movement across the membrane comes in three main flavors: passive diffusion, facilitated diffusion, and active transport.
Okay, let's break those down. Passive diffusion.
Passive diffusion is the most straightforward. It's just the movement of a substance directly across the cell membrane's oily lipid bilayer.
Straight through the wall, basically.
If it can dissolve in the oil, yes. Moving from an area of high concentration or electrochemical potential to an area of lower concentration or potential, essentially just going with the flow. No cellular energy required.
So it involves the substance dissolving in the membrane, crossing, and then redissolving on the other side.
Exactly. Partitioning and repartitioning.
Okay, then we have facilitated diffusion. What's that?
This also doesn't cost the cell any direct energy, but it relies on a transporter protein to help things along.
So it needs a helper.
Right. The transporter binds to the molecule and speeds up its movement down its electrochemical gradient. It's still a downhill journey, thermodynamically speaking, but the transporter acts like a guide, making the trip much faster than simple passive diffusion could achieve.
And it stops when things balance out.
It reaches equilibrium when the electrochemical potential is the same on both sides of the membrane. No more net movement.
Got it. Now, active transport sounds like the opposite.
It is. This is where the cell really flexes its energetic muscles. This process requires energy to move substances against their electrochemical gradient.
Pushing uphill.
Exactly, pumping them uphill. This allows the cell to create and maintain concentration differences, store potential energy, and ensure the internal environment is distinct from the outside. Really important.
And there are subtypes here.
Right. Active transport has two main subtypes: primary and secondary.
Okay, primary active transport.
Primary active transport is like using a direct energy source. It directly taps into the energy released from the breakdown, the hydrolysis of ATP adenosine triphosphate, the cell's main energy currency, to force molecules across the membrane.
So ATP is the fuel.
Directly. The ABC transporters, including the very well-known PGP, are classic examples of primary active transporters. In mammals, they generally function as unidirectional efflux pumps.
Meaning they mostly push things out of the cell.
Primarily, yes. Push things out. Another key example is the sodium-potassium ATPase, or sodium-potassium pump.
Ah, yes, the famous pump.
It uses ATP to maintain those critical sodium and potassium gradients across the cell membrane that are essential for, well, nerve impulses, muscle contraction, and many other vital processes.
Okay, so that's primary. What about secondary active transport?
Secondary active transport is a bit more clever. It's like an energy reuse system.
How so?
It harnesses the energy stored in an existing electrochemical gradient one that was typically established by a primary active transporter, like that sodium-potassium pump, to power the movement of another molecule against its own gradient.
Ah, okay.
Wow.
So it uses a pre-existing slope to push something else uphill.
Exactly. Think of it like a water wheel being driven by a stream that was initially created by an upstream pump. The energy is coupled indirectly.
Makes sense. And there are categories here, too.
Yes, secondary active transport can be further categorized into symport and antiport.
Symport.
Symport, also known as cotransport, is when the driving ion, the one moving down its gradient, and the transport molecule move across the membrane in the same direction.
Together.
Right. A prime example is the sodium-glucose cotransporter, SGLT1, in the small intestine. The inward rush of sodium moving down its steep concentration gradient literally pulls glucose along with it into the cell, even if glucose concentration is already higher inside.
Okay, so symport is same direction. Antiport must be
Opposite directions, also called exchange transport. Here, the driving ion and the transported molecule move across the membrane in opposite directions.
one goes in, one comes out.
Precisely. Our sources highlight the sodium-calcium exchanger, NCX, which is important in heart muscle cells. The influx of sodium ions moving down their electrochemical gradient fuels the expulsion, the pumping out of calcium ions from the cell.
So the cell is ingeniously using the potential energy of one gradient, the sodium gradient, to create another gradient, the calcium gradient.
Exactly. It's a very efficient way to manage ion concentrations.
This brings us to the kinetics of transport, essentially, how fast these processes happen and how things like concentration affect them.
Right, the speed and efficiency. Our source introduces the Michaelis-Menten equation, which is really fundamental in enzymology and transport biology. It describes the relationship between the rate of transport, or flux, we call that V, and the concentration of the transported molecule, the substrate, which we call S.
And the equation is V equals Vmax times S divided by Karaman plus S.
That's the one. V equals Vmax, S, Km plus S.
Okay, let's unpack this equation because it seems pretty central to understanding how these transporters actually work. Vmax.
Vmax represents the maximum rate of transport that the system can achieve when the transporter is completely saturated with substrate.
So full speed ahead.
Exactly. Think of it as a transporter working at its absolute top speed. And importantly, this Vmax is directly proportional to the number of transporter proteins you have on the cell surface. More transporters, higher Vmax.
Okay, and Karaman, the Michaelis constant.
Right. Karaman is the substrate concentration at which the transport rate is exactly half of Vmax.
It's half speed.
Right. But importantly, KLM also gives us an indication of how well the transporter binds to its substrate, its affinity. A lower column generally signifies a higher affinity.
Meaning it grabs onto its target molecule more tightly.
Yes, it doesn't take as high a concentration to get close to maximum speed if the affinity is high.
So how do we figure out these Vmax and KLM values in the lab?
Well, you measure the rate of transport experimentally at various concentrations of the substrate, then you can plot the data. One common graphical method is the Eadie-Hofstee plot.
Eadie-Hofstee. How does that work?
It's basically a rearrangement of the Michaelis-Menten equation. You plot Vs on the y-axis against V on the x-axis. If the transporter follows Michaelis-Menten kinetics, you should get a straight line.
And the line tells you.
The slope of that line is -1 km, and the y-intercept is Vmax. So it's a really useful way to visualize and quantify these key kinetic parameters of transport.
Got it. Our sources also dive into transporter inhibition. Like enzymes, these transporters can be blocked.
Absolutely. Their activity can be reduced or completely blocked by other molecules. We learn about 3 main types of inhibition: competitive, non-competitive, and uncompetitive.
Okay, competitive first. Sounds like what it is.
It is. Competitive inhibition is like having 2 keys that fit the same lock. The inhibitor molecule directly competes with the substrate for the exact same binding site on the transporter.
So if the inhibitor gets there first,
the substrate can't bind. This reduces the rate of transport at any given substrate concentration. However, if you significantly increase the concentration of the substrate, you can essentially outcompete the inhibitor.
Flood the system with the real key.
Right. So you can still reach the original Vmax eventually, but it will take a higher substrate concentration to do so. This means the apparent Kalman increases.
Okay, that makes sense. Non-competitive inhibition.
This takes a different approach. Here, the inhibitor binds to a site on the transporter that is different from the substrate binding site, what we call an allosteric site.
So it doesn't block the main spot?
Correct. This binding doesn't prevent the substrate itself from attaching to the transporter, but it interferes with the transporter's ability to actually move the substrate across the membrane. It kind of jams the mechanism.
Inhibits the translocation step.
Exactly. In this case, the Vmax is reduced because some of the transporter proteins are effectively disabled, but the kinerum, the binding affinity, remains the same because the substrate binding itself isn't directly affected.
Interesting. And the third type, uncompetitive inhibition.
This one's a bit more nuanced. The inhibitor only binds to the transporter after the substrate has already bound. It binds to the substrate transporter complex.
So it waits for the substrate to be there first.
Yes. And this binding then prevents the transporter from completing its cycle and releasing the substrate on the other side of the membrane. It traps it.
Okay, so what does that do to Vmax and kinara?
With uncompetitive inhibition, both the apparent kinara and the Vmax are reduced.
Right. Understanding these different modes of inhibition seems absolutely critical for predicting how different drugs might interact with each other by messing with transporter activity.
It's hugely important in pharmacology, yes. Predicting and avoiding unwanted drug-drug interactions.
Now that we have a solid grip on the basic mechanisms and kinetics, let's explore the structure and mechanism of these incredible protein machines. How do they actually look and work?
Great. Our sources emphasize that both SLC and ABC transporters are multi-membrane spanning proteins.
Meaning they weave back and forth across the membrane?
Exactly, multiple times. This creates the necessary binding pockets and pathways for the substrate to move through. And recent advancements in structural biology, particularly cryo-electron microscopy or cryo-EM, have given us amazing insights into their three-dimensional architecture and how they physically move their cargo.
Let's start with the ABC transporters. What do we know about them?
Well, we learned that the human genome encodes 49 genes for these proteins, and a defining feature is the presence of one or two highly conserved ABC regions.
ABC, standing for ATP binding cassette.
Precisely. These ABC regions are the core catalytic units. They bind and hydrolyze ATP, breaking it down to release energy, and that's what provides the power for the active transport we discussed earlier.
So they're the engines.
They are the engines. Interestingly, in eukaryotes like us, most ABC transporters are involved in moving compounds out of the cytoplasm, either across the main cell membrane or into intracellular compartments like the endoplasmic reticulum or mitochondria.
But in bacteria?
In bacteria, they often play a critical role in importing essential nutrients into the cell, so directionality can differ.
Okay. What about their overall structure?
The general structural organization, as revealed beautifully by cryo-EM, is often quite symmetrical. They typically consist of two main domains, the nucleotide binding domains, or NBDs.
The ATP binders?
Yes, located on the cytoplasmic side of the membrane. And two transmembrane domains, or TMDs, which are embedded within the lipid bilayer itself.
And the NBDs, the powerhouses?
They are. They contain specific, highly conserved sequence motifs like the Walker-A motif and the ABC signature motif that are directly involved in binding and then breaking down hydrolyzing ATP.
And that ATP binding and breakdown drives the whole process?
It does. When ATP molecules bind to both NBDs, it triggers a really substantial conformational change in the entire transporter protein. This leads to an outward-facing state where the substrate binding site within the TMDs becomes accessible from the outside of the cell or the lumen of an organelle.
So the gate opens outwards.
Right. Then, the subsequent hydrolysis of the bound ATP and the release of the products ADP and inorganic phosphate cause the transporter to flip back to an inward-facing conformation.
Releasing the substrate inside or picking up a new one?
Exactly. Releasing the substrate if it was imported, like in bacteria, or resetting to pick up a new substrate from the cytoplasm for export, common in humans.
And the TMDs, the transmembrane domains, what's their job?
The TMDs are the parts responsible for recognizing and specifically binding the target molecule, the substrate. And then they facilitate its movement across that tricky hydrophobic environment of the cell membrane.
And how are they structured?
It varies. In some ABC transporters, like the famous P-glycoprotein or PGP, the TMDs are formed from a single long polypeptide chain that just loops back and forth across the membrane many times.
One big protein.
Yes. But in others, like breast cancer resistance protein or BCRP, the TMDs are formed when two identical protein subunits come together to form a homodimer.
Two proteins working together.
Correct. Our source details the FLOPAZ model to explain how PGP pumps out these amphiphilic molecules, ones that have both water-loving and fat-loving parts.
How does that work?
The current thinking is that the substrate first dissolves into the inner leaflet, the inner layer of the lipid bilayer. Then it kind of diffuses sideways within the membrane until it binds to PGP.
So it binds within the membrane itself.
It seems so. Then the energy from ATP hydrolysis drives that conformational change in PGP, which essentially flips the substrate to the outer leaflet of the bilayer.
Pushes it to the other side of the membrane.
Right. From there, it can then partition out into the extracellular space. And it's generally believed that this whole cycle consumes one molecule of ATP for each molecule of drug transported.
1 ATP per customer.
Seems that way. After releasing the ATP breakdown products, the transporter resets to its inward-facing state, ready to grab another substrate from the cytoplasm and do it all over again.
And you mentioned some only have one ABC motif.
Yes, it's important to remember that some ABC transporters only contain one ADC motif per polypeptide chain, but they function either as homodimers, like BCRP, where two identical units pair up, or as heterodimers, like the ABCG5 and ABCG8 proteins involved in sterile transport, where two different subunits pair up.
Okay, that covers the ABCs. Now let's switch to the solute carrier, or SLC superfamily. You said this group is huge and diverse.
Incredibly so. This group includes not only secondary active transporters that use ion gradients, but also facilitated transporters that just function more like ion channels.
How many are there?
Our source highlights a staggering 65 families within the SLC superfamily, comprising around 458 individual transporter genes in the human genome. It's vast.
Wow. So much variety.
Yes, remarkable structural and functional diversity. But despite that, most SLC transporters share a common architectural theme. They all possess multiple transmembrane domains, typically ranging from 7 to 12 alpha helical segments that span the lipid bilayer.
Okay. Are there common structures within this diversity?
Yes. Detailed structural analyses using X-ray crystallography and cryo-EM have revealed that the transmembrane domains often exhibit a kind of pseudo-symmetrical arrangement. This suggests they might have evolved through gene duplication and fusion events over time.
Interesting.
And their overall three-dimensional protein folds can be broadly categorized into two major groups: the major facilitator superfamily, or MFS fold,
MFS fold.
and the leucine transporter, or LUT fold.
LUT fold.
Yeah.
Okay, let's start with the MFS fold. What uses that?
Many of the glucose transporters, the GLUTs, which belong to the SLC2 family and are crucial for getting glucose into our cells, exhibit this MFS fold.
Okay, any others?
Interestingly, yes. Many clinically important drug transporters within the SLC22 family, which handle a wide range of organic ions, also share this MFS fold.
How does the MFS fold work?
Proteins with this fold typically have two halves, or domains, each usually containing 6 transmembrane segments, arranged kind of like an inverted repeat. These two halves move relative to each other, acting like a rocker switch.
A rocker switch.
Yeah. The substrate binding site is located in a central cavity formed between these two halves, and the rocker switch mechanism allows this binding site to alternately be exposed first to one side of the membrane, say the outside, and then to the other side, the inside.
So it rocks back and forth, showing the binding site to each side.
Precisely. When a specific substrate molecule binds to the accessible site, let's say on the outside of the cell, it triggers a subtle but crucial shift in how the two domains are oriented relative to each other.
The rocking motion.
Right. This conformational change effectively reorients the substrate binding site, now exposing it to the other side of the membrane, maybe the cytoplasm, where the substrate can then be released.
And for this to work efficiently.
For this exchange process to function well, especially in antiporters that swap molecules, the binding of different substrates, like the incoming molecule and the outgoing one, must be mutually exclusive. There's typically only one binding site that undergoes this alternating exposure.
Got it. So that's the MFS fold and the rocker switch. What about the other one, the LUT fold?
The leucine transporter, or LUT fold, is exemplified by transporters found in the SLC5 family, which includes those sodium-glucose cotransporters we mentioned earlier, SGLTs, and the SLC6 family, which includes many important neurotransmitter transporters.
Okay, how does this one work?
This fold forms what's been described as a rocker bundle, or maybe a gated pore structure. It operates through a slightly different mechanism compared to the rocker switch.
Different how?
Here, the transporter also has two main halves, each usually containing 5 transmembrane domains. But only one of these halves seems to be primarily responsible for the alternating access movement, exposing the substrate binding site first to one side and then the other. The other half remains relatively more stationary, providing structural support.
So more like one part moving against a stable base.
That's a good way to think about it, yes. A rocker bundle rather than two equally moving parts. It's fascinating how these seemingly subtle structural differences at the molecular level can lead to distinct mechanisms for moving substrates across the membrane.
And the range of molecules these SLC transporters handle is incredibly broad, right?
Absolutely. Inorganic ions, a huge variety of organic molecules charged, uncharged, as well as a multitude of xenobiotics, including many, many clinically important drugs.
Okay, building on these mechanisms, our sources introduce the critical concept of vectorial transport. What does that mean?
Vectorial transport describes the directional or asymmetrical movement of solutes across a layer of polarized cells. Think about the epithelial cells lining your intestines, or the endothelial cells forming the blood-brain barrier. These cells have distinct sides, like an inside and an outside.
Like apical and basolateral membranes.
Exactly. Vectorial transport is about moving things specifically from one side to the other, and this directionality is absolutely essential for tons of physiological processes.
Such as?
Well, absorbing nutrients and drugs in the gut, eliminating waste products and drugs via the liver and kidneys, and creating and maintaining those specialized barrier functions that protect sensitive tissues like the brain.
How do ABC and SLC transporters fit into this?
We learned that ABC transporters, being generally unidirectional efflux pumps, inherently contribute to vectorial transport. They usually pump things out.
One-way street?
Pretty much. SLC transporters, however, can mediate either uptake or efflux, often depending on the direction of the electrochemical gradient.
So they can go both ways.
Potentially, yes. Therefore, for SLC transporters to contribute to directed transport across a polarized cell layer, they often need to work in a coordinated fashion with other transporters located on the opposite membrane of the cell.
Teamwork.
Exactly. A solute might be taken up into the cell across one membrane by an SLC transporter and then exported out across the opposite membrane by a different transporter, could be another SLC or maybe an ABC family member.
What about lipophilic compounds, things that can cross membranes easily?
Right. For those lipophilic compounds that can readily effuse across cell membranes because they dissolve in the lipid bilayer, ABC transporters alone might be sufficient to achieve vectorial transport. If the compound can passively enter the cell from one side, say the blood side, and then be actively pumped out on the other side, like into the gut lumen by an ABC transporter, you get net directional movement.
But for more hydrophilic things, water-loving ions.
For those, achieving efficient directed movement across a polarized cell layer often requires the concerted action of both uptake and efflux transporters, strategically located on the different plasma membrane domains of the cell.
A coordinated system.
Definitely. A common strategy involves having a primary or secondary active transporter on one membrane, say, the basolateral membrane facing the bloodstream that actively pulls the solute into the cell, maybe against its gradient.
Okay, loaded up.
Then a passive or facilitated transporter located on the opposite membrane, say, the apical membrane facing the lumen of the intestine or kidney tubule, allows the solute to exit the cell, now moving down the concentration gradient that was created by the first transporter.
So pump it in one side, let it flow out the other.
That's the essence of it. This coordinated action ensures efficient transcellular movement of the compound across the entire epithelial layer.
Where do we see good examples of this?
The liver provides an excellent and really well-studied example of this elegantly coordinated transport system.
Okay, tell me about the liver.
On the sinusoidal membrane of hepatocytes, that's the side facing the blood, these liver cells express a variety of SLC uptake transporters. You've got the OATPs, organic anion transporting polypeptides, OCTs, organic cation transporters, and NTCP, the sodium teroxalate co-transporting polypeptide.
And they pull stuff in from the blood.
Exactly. They import a wide range of substrates, bile acids, organic anions, organic cations from the circulating blood into the liver cells.
Then what happens?
Then, on the canalicular membrane, that's the side facing the tiny bile ducts, hepatocytes express several ABC efflux transporters. We're talking MDR1, MRP2, BCRP, BSF, the bile sol export pump, and MDR3.
And these pump things out into the bile.
Right. They actively transport these imported compounds, plus their metabolites formed in the liver, and various endogenous substances like bile salts and phospholipids into the bile for eventual elimination from the body.
So it's like a highly organized directional conveyor belt system at the cellular level.
That's a perfect description. Our source even mentions studies using doubly transfected cells.
What are those?
Researchers engineer cells in the lab to simultaneously express both an uptake transporter on one surface and an efflux transporter on the opposite surface. These experiments provide really compelling evidence for efficient transcellular transport when these transporters work together.
And we see similar systems elsewhere.
Absolutely. Similar sophisticated and coordinated systems of uptake and efflux transporters are found in other key places, like the epithelial cells lining the intestine, the cells of the renal tubules in the kidney, and the endothelial cells of the brain capillaries forming the blood-brain barrier.
Now, is the amount of these transporters fixed, or can the cell control how many are present?
That's a great question. No, their expression levels and activity are not static at all. They can be dynamically regulated at the transcriptional level, controlling how much mRNA is made from the gene.
What triggers these changes?
A variety of physiological and pharmacological stimuli, things like exposure to drugs, hormones, and even changes in your metabolic state can alter transporter expression.
How does that regulation happen?
A key mechanism involves a class of proteins called type 2 nuclear receptors. These include important players like PXR, pregnant X receptor, CRR, constitutive androstenedione receptor, FXR, farnesoid X receptor, PPRI, peroxisome proliferator activated receptor alpha, and RAR, retinoic acid receptor.
Nuclear receptors. They sense things and control genes.
Exactly. They often function by forming pairs, heterodimers, with another nuclear receptor called RXR, retinoid X receptor. This receptor pair then binds to specific DNA sequences called response elements, located near their target genes, in this case, transporter genes.
And that binding turns the gene expression up or down.
It influences the rate of transcription, yes,
Yeah.
usually turning it up. With the exception of CR, which has a sort of baseline activity, the other nuclear receptors in this group are generally ligand activated.
Meaning they need something to bind to them to switch them on.
Correct. They need to bind to a specific molecule of their ligand to become activated and affect gene transcription. CIR is a bit different. It has some activity already, which can be suppressed by certain molecules or induced by others, like the drug phenobarbital.
PXR seems particularly interesting.
It is. PXR, also called SXR in humans, is notable because it has such a broad range of activating ligands. It can be switched on by various steroid hormones, bile acids, and numerous clinically used drugs, like the antibiotic rifampicin.
Does it work the same in animals and humans?
That's a crucial point. The potency with which different compounds activate PXR can vary significantly between species. So findings from preclinical studies in animal models might not always directly translate to what happens in humans. You have to be careful there.
And PXR is linked to drug metabolism enzymes too, right?
Yes, there's a well-established link between PXR activation and the upregulation of both cytochrome P450-3A4, CYP3A4, a major drug metabolizing enzyme, and P-glycoprotein, PGP, that key ABC efflux transporter.
So it ramps up both breakdown and pumping out.
It seems like a coordinated defense mechanism. PXR binds to regulatory elements in the genes for both CYP3A4 and PGP, increasing expression of both. It looks designed to enhance the detoxification and elimination of potentially harmful foreign compounds.
But it affects transporters beyond PGP.
Well, more recent research in human liver cells suggests that while PXR activation robustly induces CYP enzymes, its effects on the expression levels of certain SLC and ABC transporters might be more modest or selective than initially thought. Table 4.1 in our source actually provides a detailed summary of how activating these various nuclear receptors with different drugs affects specific transporter expression in the liver.
Beyond these nuclear receptors, are there other ways transporter expression is controlled?
Yes, epigenetic mechanisms also play a role, particularly DNA methylation.
Methylation, adding chemical tags to the DNA.
Right, adding a methyl group to cytosine bases in the DNA. This can lead to gene silencing in tissues where a particular transporter isn't needed, helping create those tissue-specific expression patterns.
So it helps turn genes off where they shouldn't be on.
Exactly. Conversely, the binding of active transcription factors can prevent this methylation and help keep the genes switched on in tissues where the transporter is required.
Which transporters are affected by this?
Several important drug transporters in the liver and kidney are known to be subject to this kind of epigenetic regulation. This includes OATP1B1, OATP1B3, OCT2, OAT1, and OAT3.
And others.
Furthermore, in various cell line models, it's been shown that the expression of MDR1, PGP, and BCRP from the ABC superfamily, as well as OCT1, OCT3, and OCTN2 from the SLC superfamily, can also be modulated by epigenetic mechanisms involving DNA methylation, so it's quite widespread.
Okay, let's dive into the specifics of these two major transporter superfamilies now, starting with the solute carrier or SLC superfamily. You already know it's huge.
It really is. As we touched on, 65 distinct families, around 458 individual genes in humans. These genes encode this vast array of membrane-spanning proteins, and critically, mutations in many of these SLC genes have been directly linked to various human genetic diseases.
Where can we see examples?
Table 4.2 in our source gives a great overview, highlighting examples relevant to pharmacology. You see families like SLC5, which includes those SGLTs targeted by diabetes drugs.
Right.
The SLC6 family, encoding neurotransmitter transporters targeted by antidepressants. And the SLC22 family, which covers a large number of organic ion transporters critical for drug absorption, distribution, and elimination.
And the table lists diseases too.
Yes. It links these families to associated genetic diseases, things like glutamate galactose malabsorption, primary systemic carnitine deficiency, Rotor syndrome, GILMAN syndrome. It really underscores the profound physiological importance of these transporters. Some rare, some more common.
What's striking of the SLCs is the sheer diversity of molecules they transport, their substrates.
It's incredible. Some SLC transporters are highly specific, only interacting with structurally similar molecules, like the SLC6 family members that recognize recognize specific neurotransmitters.
Picky eaters.
You could say that. But others, like the SLC22 organic ion transporters, are much more promiscuous. They can handle a broad range of chemically diverse compounds, endogenous metabolites, lots of different drugs, both anions and dications.
And how do they work energetically? Mostly passive.
Unlike the ABC transporters that directly burn ATP, the majority of SLC transporters are facilitative. They just help substrates move down their existing electrochemical gradient, no direct energy needed.
But some are active.
Yes. It's important to remember that some SLC transporters are secondary active transporters. They use the energy stored in ion gradients, usually sodium or proton gradients, to move their substrates uphill against their concentration gradients.
And this field is still growing?
Absolutely. Our source notes that since the previous edition, 13 new SLC families with 63 additional genes have been identified. The discovery process is ongoing. There's still a lot to learn.
Physiologically, these SLCs seem to do everything.
Pretty much. They perform incredibly diverse and absolutely vital roles. For instance, families like SLC1, 3, 6, 7, 25, and 36 are key for transporting amino acids' essential building blocks, but also important signaling molecules and energy sources.
What else?
Glucose and sugar transport relies on SLC2, 5, and 50 families. Metal transport things like zinc and iron involves SLC11, 30, 39, and 40. Even vitamin transport depends on SLC19, 46, and 52 families.
And neurotransmitters.
Right. SLC6 is crucial for neurotransmitter reuptake in the brain, and SLC18 handles packaging them into vesicles. They are everywhere doing critical jobs.
And from a drug perspective, pharmacology.
Immense importance. They're directly involved in absorbing oral drugs from the gut, eliminating drugs and metabolites via kidney and liver, and distributing drugs to target tissues.
And drug interactions.
Critically, yes.
Yeah.
SLC transporters are major players in clinically significant drug interactions. One drug affects the transport of another, potentially altering efficacy or causing toxicity. The SLCO family, encoding OATPs, interacts with drugs like statins and antidiabetics. The SLC22 family handles renal secretion of many anionic and cationic drugs, like antibiotics and antivirals. Interactions here are common.
Can we target SLCs with drugs?
Increasingly, yes. They are promising therapeutic targets. Over 100 SLC transporters are linked to monogenic disorders, making them potential targets for new treatments.
Like screening for defects.
Exactly. Some disorders caused by SLC mutations, like primary systemic carnitine deficiency due to SLC22A5 mutations, are now even detected through newborn screening, allowing early intervention.
And links to common diseases.
Yes, large-scale genome studies, GWAs, have found numerous SNPs genetic variations in SLC genes associated with increased risk for common diseases, like SLC30A8 and type 1 diabetes risk, or SLC22A45 and inflammatory bowel disease.
But you said many are still mysterious.
It's quite remarkable, but yes. It's estimated that around 30% of human SLC transporters are still considered orphans.
Meaning we don't know what they actually transport.
Yeah. Exactly. We haven't definitively identified their main physiological substrates or fully figured out their roles. A huge amount of ongoing research is focused on deorphanizing these transporters.
Any recent successes?
Yes, there have been some notable ones. SLC22A24 was identified as a transporter for steroid conjugates important for hormone balance. And SLC22A15 turned out to be a zwitterion transporter handling ergothioneine, an antioxidant, but also the drug gabapentin.
So progress is being made.
Definitely. Continuous progress in understanding the full scope of this vital superfamily.
Okay, let's switch gears to the other major player, the ATP binding cassette, or ABC superfamily.
Right. As our source details, the 7 subfamilies of human ABC transporters are also absolutely essential for a huge range of cellular processes. Lipid transport, peptide transport, and of course the efflux of drugs and xenobiotics.
And mutations cause diseases here too.
Yes, mutations in at least 13 different ABC transporter genes are known to cause or significantly contribute to various human genetic disorders. Table 4.3 gives a comprehensive list.
Beyond their famous role in multi-drug resistance, what's their main job pharmacologically?
A key aspect is the export of xenobiotics, those foreign compounds from healthy tissues. It's a crucial detoxification role. Key players in overall drug disposition include MDR1, ABCB1, P-glycoprotein, MRP2, ABCC2, and BCRP-BCG2.
Where are these located? Does it matter?
Location is everything for function. Table 4-4 in our source provides a really valuable map of where these pharmacologically important ABC transporters are found in major tissues involved in drug handling.
Like the intestine.
Exactly. In the intestine, MDR1, MRP2, and BCRP sit on the apical membrane of the epithelial cells, the side facing the gut lumen. They act as efflux pumps, pushing many oral drugs back into the gut, limiting absorption.
And other transporters are on the other side.
Right. MRP3, for instance, is found on the basolateral membrane of those same intestinal cells, the side facing the blood.
What about elimination in the kidney and liver?
They're strategically positioned there too. In the kidney, MDR1, MRP2, BCRP, and MRP4 are on the brush border membrane of the proximal tubule cells, pump compounds into the urine.
And liver.
In the liver, MDR1, MRP2, and BCRP are on the bile canalicular membrane, the side facing the bile duct, mediating secretion into bile. But on the opposite side, the sinusoidal membrane facing the blood, you find MRP3 and MRP4, which can transport certain compounds back into the blood.
They're also in barriers, like the blood-brain barrier.
Critical roles there. MDR1 and MRP4 are on the luminal blood-facing side of the BBB endothelial cells, helping keep things out of the brain. MRP1 and MRP4 are in the choroid plexus, part of the blood-CSF barrier.
Other barriers.
Blood-testis barrier involves MRP1 and MDR1. The blood-placenta barrier uses MDR1, MRP2, and BCRP on the maternal side and MRP1 on the fetal side, regulating transfer. And interestingly, BCRP gets highly ramped up in the mammary gland during lactation.
Let's talk about the MRP family specifically. What do they transport?
The multi-drug resistance-associated protein, or MRP family, primarily transports organic anions, as detailed in Table 4.4. MRP1 and MRP2 handle a wide range conjugates with glutathione, gluturonide, sulfate, also non-conjugated amphipathic anions, and even some neutral or cationic anti-cancer drugs, possibly co-transported with glutathione.
What about MRP3?
MRT3 has a similar substrate range to MRP2, but maybe lower affinity for glutathione conjugates. It's mainly on the sinusoidal side of liver cells and gets induced in cholestasis blocked bile flow, where it helps return potentially toxic bile salts and bilirubin glucuronides back to the blood.
And MRP4?
MRP4 handles a diverse array of negatively charged molecules. Cytotoxic drugs like 6-mercaptopurine and methotrexate, cyclic nucleotides, antivirals like adefovir and tenofovir, even some diuretics and cephalosporins. And glutathione can enable it to transport things like terauchiate and leukotriene B4.
MRP5 and MRP6?
MRP5 seems to have a narrower range, mainly nucleotide analogs and anti-HIV drugs. MRP6 is linked to a disease called pseudoxanthoma elasticum, but its specific substrate or the exact mechanism causing the disease is still largely unknown.
Okay, what about BCRP? Breast cancer resistance protein.
BCRP, also known as ABCG2, handles both neutral and negatively charged molecules. Substrates include cytotoxic drugs like topotecan and flavopiridol, methotrexate, sulfated conjugates like estrogen sulfate, antibiotics like nitrofurantoin and fluoroquinolones, statins like pitavastatin and rosuvastatin.
A wide range, too.
Yes, and even some food toxins like phytoestrogens and certain carcinogens. Plus, genetic variations in BCRP are linked to hyperuricemia and gout, and it plays a role in handling uric acid itself and gout medications like allopurinol and oxypurinol.
How do we know about their normal physiological roles?
A lot comes from studies using knockout animals in mice bred without the gene, and from studying patients with inherited genetic defects in these transporters. Many clearly play key roles in handling xenobiotics.
What happens if they're missing?
Often, mice lacking genes like MDR1, MINSRP1, MISRP4, BCRP, or BSEP are viable and fertile under normal conditions. They might just show increased sensitivity to certain drugs or toxins.
So they're mainly for defense.
It seems that way for many involved in drug disposition. The key takeaway is that while knocking out some of these might not cause obvious problems without a chemical challenge, inhibiting the function of those ABC transporters that do have critical endogenous roles, especially those linked to the diseases in Table 4.3 through drug interactions, is something we definitely want to avoid due to the risk of serious side effects.
Clinically, MDR1 or P-glycoprotein seems really prominent.
It really stands out, probably the most studied and clinically relevant ABC transporter. We learned that oral digoxin bioavailability can be significantly reduced by rifampin, which induces MDR1 expression.
Makes sense. More pump means less drug absorbed.
Exactly. And conversely, MDR1 inhibitors drugs like quinidine, verapamil, ritonavir reduce the renal excretion of digoxin because MDR1 is also on the kidney tubule brush border, pumping it into urine.
So interactions are a big deal, especially for certain drugs.
Absolutely, particularly for drugs with a narrow therapeutic window, where the effective dose is close to the toxic dose. Think digoxin, cyclosporine, tacrolimus. You have to be really careful about MDR1 interactions with these.
What about MRP3 clinically?
MRP3 plays a role in drug absorption in the intestine, often working with uptake transporters. In the liver, being on the sinusoidal side, it fluxes drugs back into the blood. This can decrease biliary excretion efficiency and maybe shorten the half-life of some drugs or their metabolites.
And MRP4?
MRP4 substrates can also be handled by OAT1 and OAT3 on the kidney's basolateral side. It seems transport across the luminal membrane by MRP4 might be the rate-limiting step for secretion. So MRP4 dysfunction might increase drug concentration inside kidney cells but have less effect on overall blood levels.
Let's revisit BCRP and drug interactions. You mentioned rosuvastatin.
Right. Intestinal BCRP limits oral absorption by pumping drugs back into the gut. It's involved in many interactions. A classic example is rosuvastatin and cyclosporine. Rosuvastatin is a substrate for both BCRP, efflux in gut liver, and OATP1B1, uptake in liver. Cyclosporine inhibits both.
So that's complicated.
Very. Cyclosporine inhibits BCRP in the gut, increasing rosuvastatin absorption. But it also inhibits OATP1B1 in the liver, decreasing uptake into the liver where it works and potentially increasing blood levels further. The net effect on systemic levels and risk of muscle toxicity depends on the balance of these opposing effects.
And the effect on cholesterol lowering depends on the liver concentration.
Precisely. It highlights the complexity of predicting the outcome of multi-transporter interactions.
Moving to the broader picture, our sources stress how vital transporters are for pharmacokinetics, what the body does to the drug.
Absolutely. Absorption, distribution, metabolism, excretion transporters are key players, especially in the liver and kidney for removing drugs from the blood, influencing metabolism and excretion. Figure 4.1 and Table 4.4 really illustrate this well.
Let's focus on hepatic transporters first. The liver.
Okay. On the sinusoidal membrane facing the blood, you have SLC uptake transporters pulling things into the liver cells, OATPs for organic anions like bilirubin and many drugs, OCTs for cortications, and NTCP for bile salts using sodium gradient energy.
Pulling things in, then what?
Then, on the opposite side, the canalicular membrane facing the bile duct, you have ABC efflux transporters pumping things out into the bile, MRP2, MDR1, BCRP, BSEP, MDR3 pumping drugs, metabolites, bile salts, phospholipids, often against a gradient, using ATP.
This coordinated uptake and efflux, this vectorial transport, that's key for liver function.
Absolutely essential. It determines the overall exposure of a drug both in the blood and within the liver tissue itself. Figure 4-9 gives a great overview of these transporters, and this interplay is fundamental to understanding many clinically significant drug interactions. Our sources give several good examples.
Like statins.
Yes. HMG-CoA reductase inhibitors, the statins. Most active forms are substrates for hepatic uptake, especially OATP1B1. They also undergo enterohepatic recirculation excreted in bile, reabsorbed in gut.
Why is OATP1B1 uptake so important for statins?
It's crucial for concentrating the drug in the liver, where it inhibits cholesterol synthesis. This targeted delivery helps minimize systemic levels, reducing the risk of muscle side effects like myopathy.
And genetics matters here.
Hugely. Genetic polymorphisms in the OATP1B1 gene, SLCO1B1, significantly affect its function, leading to big differences between people in statin efficacy and their risk of side effects.
What about antivirals, like for hepatitis C?
Another great example, several HCV, NS34A, protease inhibitors, simiprevir, glycoprevir, grazoprevir, show nonlinear pharmacokinetics. Their elimination doesn't increase proportionally with the dose.
Why not?
Partly because their hepatic uptake via OATP1B transporters get saturated. The transporters can only work so fast.
So if you add an OATP1B inhibitor,
if you co-administer them with OATP1B inhibitors like rifampicin or cyclosporin A, their systemic exposure can shoot way up. Potential for more efficacy, but also higher risk of side effects. It really highlights OATP1B's role in interactions.
and other HCV drugs.
Interestingly, while the NS5A and NS5B inhibitors aren't usually OATP1B substrates themselves, some can inhibit OATP1B activity, potentially causing interactions with other drugs that are OATP1B substrates.
Let's talk about gemfibrozil and statins. That interaction is infamous.
It is. Gemfibrozil, used to lower triglycerides, increases the risk of statin-induced myopathy through multiple mechanisms. One key way is by inhibiting OATP1B1 uptake of statins into the liver, affecting both the parent drug and its glucuronide metabolite. metabolite. Gemperberzel also inhibits CYP2C8, an enzyme that metabolizes some statins. Both effects increase statin plasma levels and muscle toxicity risk.
But not all fibrates are the same.
Correct. Pemafibrate, another drug in the class, seems safer with statins, possibly due to higher selectivity and being effective at a much lower dose. Although pemafibrate itself is an OATP1B-CYP substrate, its exposure can increase greatly if given with OATP1B inhibitors like rifampicin or cyclosporin A.
Irinotecan, the cancer drug.
Yes, CPT11. It's a prodrug converted to the active metabolite SN38. Both SN38 and its glucuronide conjugate are taken up by OATP1B in the liver and excreted into bile via the efflux transporter MRP2.
And this causes problems.
This significant biliary excretion of the active metabolite often leads to severe gastrointestinal toxicity, especially diarrhea. Preclinical studies show that inhibiting MRP2 with probenecid reduced diarrhea, suggesting a potential way to manage this side effect.
Bosentan for pulmonary hypertension.
Another drug significantly influenced by hepatic transporters. It's taken up by OATP1B1 and OATP1B3, then metabolized by CYP2C9-3A4. Uptake is key for its elimination.
So inhibitors affect it.
Yes, co-administration with OATP1B inhibitors like cyclosporine or rifampicin affects its pharmacokinetics. Plus, its OATP1B uptake can saturate at therapeutic doses, leading to nonlinear kinetics.
What about ACE inhibitors like timocaprol?
Timocaprol is interesting. Its active metabolite, timocaprolat, is eliminated by both liver, bile, and kidney urine. Most other ACE inhibitors are mainly renal.
So kidney problems affect it less.
Exactly. Plasma levels are less affected by renal failure. Timocaprolat is a substrate for both OATPs and MRP2. Other ACE inhibitors aren't usually good MRP2 substrates, even if they use OATPs for uptake.
So MRP2 affinity might determine how much gets into bile.
That's the hypothesis. It might explain why timocaprolat has dual excretion. And perhaps drugs excreted equally by bile and urine might show less pharmacokinetic variability between individuals.
Angiotensin II receptor antagonists like valsartan.
Hepatic uptake and biliary excretion are important for them too. Telmisartan uses OATP1B3 mainly. Valsartan and olmesartan use OATP1B1 and OATP1B3. And MRP2 seems key for their biliary excretion.
Finally, antidiabetic drugs like repaglinide.
Yes, the meglitinides. They're mainly metabolized by CYPs, but their elimination rate is also significantly influenced by hepatic uptake via OATPs, particularly OATP1B1.
So genetics matters here too.
Definitely. SLCO1B1 polymorphisms variations in the OATP1B1 gene significantly affect repaglinide pharmacokinetics. And the common SLCO1B1 521TC variant alters nataglinide and glibenclamide kinetics too. OATP1B1 is a key determinant, even though metabolism is the primary clearance route.
Okay, let's shift focus now to the renal transporters in the kidney, critical for excretion. Let's start with organic concations.
Right. The proximal tubule of the nephron is a major site for actively secreting a huge variety of organic cations into the urine.
What kind of cations?
Both endogenous ones our body makes, like choline, N-methylnicotinamide, dopamine, and lots of foreign compounds, including many positively charged drugs like cimetidine, ranitidine, metformin, varenicline, trospium, nicotine, even toxins like paraquat. They vary from hydrophilic to hydrophobic, often under 400 Daltons. Figure 410 gives a nice visual model.
How do they get secreted? 2 steps.
Yes, sequentially across 2 membranes of the kidney tubule cells. First, across the basalaral membrane, from the blood side into the cell, then across the apical membrane from the cell into the tubular lumen, the urine side.
Basalateral uptake first. Which transporters?
In humans, it seems organic cations cross the basalateral membrane mainly via 2 SLC22 family members, OCT2, organic cation transporter 2, and OCT3, organic cation transporter 3. This uptake usually happens down the electrochemical gradient.
Then apical efflux into the urine. How?
This step is largely mediated by an electroneutral proton-organic cation exchange. This is carried out by transporters from the SLC47 family, also known as the MATI family multi-drug and toxin excursion.
MATI transporters.
Key members MATN1 and MAT2K are on the apical membrane and pump hydrophilic cations out. Additionally, the OCTN subfamily OCTN1, SLC22A4, and OCTN2 SLC22A5, also on the apical side, contribute to this efflux.
OCTNs are involved too.
Yes, and they're interesting because they're bifunctional. They secrete carnitines, but also reabsorb carnitine from the urine back into the cell using sodium cotransport. In their secretory mode, though, they seem to act as proton-organic cation exchangers, protons in, cations out.
Where do the protons come from?
That inward proton gradient is likely maintained by sodium-hydrogen exchangers, NHEs, from the SLC9 family, which themselves depend on the sodium gradient set up by the main NK-ATP on the basolateral side.
Which step is slowest?
It's generally thought that the transport across the luminal membrane via mates and possibly OCTNs is often the rate-limiting step in overall renal cation secretion.
Can you tell us more about specific OCTs and mates, OCT2?
OCT2 is highly abundant in the human kidney proximal distal tubules, collecting ducts, specifically basolateral in the proximal part. It mediates electrogenic transport of things like MPPO and TEA, can do cation exchange, has a broad substrate range for monovalent concentrations under 400 day, but low affinity for monoamine neurotransmitters. OCT3 is more widespread liver, kidney, less than OCT12 there, intestine, placenta, muscle, fat. Also, potential sensitive transport plays a role in renal elimination and intestinal absorption of metformin.
OCTN1.
OCTN1 seems to act as an organic cation proton exchanger. Influx better at alkaline pH, efflux with an inward proton gradient, has a nucleotide binding motif, ATP stimulates it, can also do cation exchange, important for renal transport of gabapentin.
OCTN2.
OCTN2 is that bifunctional one. Non-dependent carnitine uptake and non-independent organic cation transport. Its cation transport is pH sensitive, might be a cation exchanger. Carnitine transport is electrogenic. Mutations cause primary systemic carnitine deficiency due to impaired renal carnitine reabsorption.
And mates, MATE1 and MATE2K.
They interact with diverse hydrophilic metformin, cimetidine, topotecan. Also some anions like acyclovir, ganciclovir. Even zwitterians like cephalexin and cephridine, maybe one specific. The herbicide paraquat has high affinity for MATE1.
Where are they?
Both are apical in the proximal tubule. MATE1 is also in the liver's canalicular membrane. They act as apical organic cation proton antiporters. Proton gradient drives kinnic E-chiaflux. Interestingly, antibiotics like levofloxacin and ciprofloxacin are potent inhibitors but aren't transported.
What about genetic variations in these?
OCT1 has the most known polymorphisms, then OCT2, OCT3. Variants in OCT12 are linked to altered metformin elimination and response. Mates have fewer polymorphisms, but non-coding variants in the MATE-E genes, SLC47A12, are also linked to metformin response variation.
Okay, let's switch to organic anion transport in the kidney. What's its main job?
Primarily xenobiotic removal, handling structurally diverse, weakly acidic drugs, pravastatin, captopril, PAH, penicillins, and toxins like ochratoxin. OATs, organic anion transporters, can move both hydrophobic and hydrophilic anions and might interact with cations or neutral compounds too. Figure 411 gives a model.
How's it work? Basolateral uptake first.
Yes. Uptake from blood into the kidney cell across the basolateral membrane is mainly via two SLC22 members, OAT1, SLC22A6, and OAT3, SLC22A8. This uptake occurs against the electrochemical gradient for many anions.
How is it powered?
It's an indirect mechanism. It involves exchanging the intracellular organic anion for an intracellular molecule called alpha-ketoglutarate, which then moves down its gradient out of the cell into the blood.
Where does the alpha-ketoglutarate gradient come from?
That outward gradient is maintained partly by a basolateral sodium dicarboxylate cotransporter, NADC3. It uses the sodium gradient from the NACA-IPase to pull dicarboxylates like alpha-ketoglutarate into the cell from the blood. So ultimately, the sodium gradient powers organic anion uptake.
Clever. Secondary active transport again, then efflux into the urine.
The mechanism for apical efflux is still a bit controversial, actually. OAT4 might play a role, but it's also stimulated by alpha-ketoglutarate exchange, suggesting it might be more involved in reabsorption from the urine back into the cell.
Other candidates.
NaPT1, the phosphate transporter, can transport some hydrophilic anions like PAH with low affinity, maybe contributing to efflux. And the ABC transporters, MRP2 and MRP4, are also on the apical membrane and can actively pump some organic anions out.
Tell us about specific OATs. OAT1?
OAT1 is mainly kidney, some brain muscle, basolateral in the S2 segment of the proximal tubule, handles saturable pH transport. Activity is transstimulated by other anions, including alpha-ketoglutarate, transports low MW organic anions like PGE2, urate, drugs, toxins. Also some neutral compounds like cimetidine with lower affinity, expression regulated by sex hormones.
OAT2.
OAT2 is in kidney and liver, basolateral renal proximal, bidirectional facilitative transporter for nucleotides, especially CGMP, low affinity for PA methotrexate, high for PGE2, lower for some neutrals like cimetidine.
OAT3.
OAT3 in humans is mainly basolateral proximal tubule. Two variants exist. One transports a wide range of anions, PAH, estrone sulfate, pravastatin, cimetidine 6MP, methotrexate. The other seems inactive. Specificity overlaps OAT1, but kinetics differ. Estrone sulfate higher affinity for OAT3, cimetidine higher for OAT1. OAT4.
OAT4 is in placenta and luminal proximal tubule in the kidney. Transport stimulated by alpha-ketoglutarate gradients. May be involved in reabsorption, not secretion. Substrates include estrone sulfate, PAH, zidovudine, tetracycline, methotrexate.
Are there other important anion transporters in the kidney?
Yes. Your AT1, SLC22A12, is kidney-specific, apical proximal, mainly reabsorbs urate back into blood, electroneutral, stimulated by chloride.
NPT1.
NPT1, SLC17A1, is luminal proximal and in brain. Transports PAH, probenecid, penicillin G, May efflux anions. Interacts with uric acid too. Figure 42-2 details renal urate transport.
And the ABC transporters MRP2 and MRP4.
MRP2, ABCC2, is known for effluxing drug conjugates into bile, but it's also apical in the kidney proximal tubule, effluxing organic anions, usually larger ones than OBATs handle. MRP4, ABCC4, is also apical renal, handles conjugated anions, glucuronide DSH, interacts with methotrexate, antivirals, cyclic nucleotides.
BCRP, ABCG2, is also apical renal and duodenum, secretes uric acid, and the gout drugs allopurinol.
Any polymorphisms here?
OAT1, OAT3 have polymorphisms in different ethnic groups. Check pharmgkb.org. And ABCG2 polymorphisms are linked to reduced response to allopurinol.
Okay, let's shift from kinetics to dynamics. How are transporters involved in pharmacodynamics? How drugs actually work in the body? Let's start with the brain.
Right. Think about biogenic amine neurotransmitters. Norepinephrine, NE, serotonin, 5-HT, dopamine, DA. They're stored in vesicles in presynaptic neurons, released into the synapse, signal to the next neuron, and then their action needs to be stopped.
How is it stopped? Reuptake.
A major way is reuptake back into the pre- or postsynaptic cells. And the plasma membrane transporters responsible for this neuronal reuptake, regulating levels in the synaptic cleft, primarily belong to the SLC1 and SLC6 superfamilies.
Neurotransmitters.
They handle reuptake of GABA, glutamate, NE, 5-HT, DA, the major ones. And critically, these transporters themselves are often the pharmacological targets for many neuropsychiatric drugs.
Like antidepressants.
Exactly. Antidepressants, drugs for anxiety, ADHD, and others often work by modulating these SLC transporters.
Which specific SLC6 members are key in the brain?
For monoamine reuptake into presynaptic neurons, it's NET, norepinephrine transporter, SLC6A2, DAT, dopamine transporter, SLC6A3, and SERT, serotonin transporter, SLC6A4. For GABA, it's the GATs, GAT123.
And they all look similar structurally.
Generally, yes. They typically have 12 transmembrane regions and a large glycosylated extracellular loop between TM3 and TM4.
How are they powered?
They're secondary active transporters. They use the energy of the sodium Na-U gradient to drive neurotransmitter uptake. Chloride, Cl, is often required too.
So their job is to clear the synapse.
Right. They regulate the concentration and how long the neurotransmitter hangs around in the synaptic cleft by pulling it back into the neuron. This terminates the signal.
Does this affect storage?
Yes. The efficiency of reuptake influences how much neurotransmitter is available for repackaging into synaptic vesicles for the next release. That packaging step involves vesicular transporters from the SLC17 glutamate and SLC18 monoamines families.
Are these brain-only transporters?
No, many SLC6, 17, and 18 members are also found elsewhere, intestine, kidney, platelets doing other jobs. And interestingly, some plasma membrane transporters can even run in reverse under certain conditions, exporting neurotransmitters without needing sodium.
Let's focus on GABA uptake. Which GAT is most important?
GAT1, SLC6A1, is probably the most important in the brain. Found in GABAergic presynaptic neurons, abundant in neocortex, cerebellum, basal ganglia, etc., key for regulating GABA signaling.
What about the others? GT3?
GAT3, SLC6A11 is also in the brain, but mainly in glial cells, not neurons. GT2 SLC6A13 is more peripheral, kidney, liver, but also in the CNS choroid plexus meninges, maybe helps regulate GABA in the cerebrospinal fluid. BGT1 SLC6A12 is found in extrasynaptic areas like hippocampus cortex.
Is GABA on drug target?
Yes, it's the target of the anti-epileptic drug tiagabine. By inhibiting GABA1 reuptake, tiagabine prolongs GABA's inhibitory effects in the synapse.
Okay, catecholamines. Norepinephrine.
That's handled by NET. SLC6A2, found in central and peripheral nervous tissue, adrenal glands, reuptakes NE and some DA into neurons, limiting synaptic duration, terminating action, salvaging neurotransmitter for reuse.
Drug targets for NET.
Yes. Antidepressants like disipramine and other tricyclics, cocaine, and the SNRIs, serotonin, norepinephrine reuptake inhibitors like duloxetine and venlafaxine, which also hits SIRT.
Any diseases linked to NET?
Yes, mutations in NET have been linked to orthostatic intolerance, affecting blood pressure regulation.
Dopamine uptake.
That's primarily DA, SLC6A3, mainly in brain dopaminergic neurons. Reuptakes DA, terminates its action, found at the synapse, but also along the neuron away from the cleft.
What does date influence?
Mood, behavior, reward, cognition. It interacts with cocaine, amphetamines, and the neurotoxin MPTP, which causes Parkinson's-like symptoms by getting into dopamine neurons via date.
Serotonin uptake.
That's SIRT, SLC6A4. Reuptakes and clears serotonin, 5-HT in the brain. Needs NAO and CLA, maybe countertransports call.
What does SIRT transport besides serotonin?
Tryptamine derivatives, neurotoxins like MDMA, ecstasy, and phenfluramine.
And drug targets.
It's the specific target of SSRI antidepressants, fluoxetine, paroxetine. Also one target of tricyclic antidepressants like amitriptyline.
Genetics matter for SIRT.
Yes, SIRT gene variants are linked to behavioral neurological disorders like depression and anxiety. But exactly how reduced SIRT activity from genetics or drugs affects behavior isn't fully understood. It's complex.
Let's shift to another area where transporters are key pharmacodynamic players.
Yeah.
Antidiabetic drugs, especially those targeting glucose.
Right. Glucose needs transporters to cross membranes, and glucose homeostasis is mainly controlled by two SLC families, SLC2, facilitated GLETs, and SLC5, sodium glucose cotransporters, SGLTs.
Where are the SGLTs?
SGLT1, SLC5A1, is in the intestine for glucose absorption from food and also in the kidney for reabsorption. SGLT2, SLC5A2, is primarily in the kidney's proximal tubule. It does almost all the glucose reabsorption there.
What happens if these are mutated?
Mutations inactivating SGLT2 cause profound glucosuria, lots of glucose in the urine. But mutations in SGLT1 usually have milder effects on overall glucose levels.
And this led to new drugs.
Exactly. This observation that SGLT2 is the main player in renal glucose reabsorption led directly to developing SGLT2 inhibitors, the gliflozins like canagliflozin, dapagliflozin, empagliflozin, for type 2 diabetes.
How do they work?
They specifically inhibit SGLT2 in the kidney. This reduces glucose reabsorption from the renal tubules back into the blood. More glucose gets excreted in the urine, lowering systemic blood glucose levels.
Why is that good for type 2 diabetes?
Because in type 2 diabetes, SGLT2 expression and glucose reabsorption are often increased. So inhibiting it helps counteract that.
Do they have other benefits? Kidney protection.
Yes, inhibiting glucose reabsorption and maybe affecting glucose metabolism within the kidney might contribute to their benefits on diabetic nephropathy, though the full mechanisms are still being worked out. They're also now used for certain types of heart failure.
Let's move to a really critical interface. The blood-brain barrier, the BBB. How do transporters feature here pharmacologically?
The BBB is this highly specialized, tightly regulated barrier protecting the central nervous system, CNS. It keeps out circulating neurotransmitters, toxins, bacteria, while letting in essential nutrients and ions. Chapter 17 has more detail, but transporters are key.
How is the barrier formed physically?
Mainly by the specialized endothelial cells lining the brain's microvasculature. They have tight junctions between them, severely limiting stuff leaking through the gaps, paracellular flow. They also have very low vesicular transport compared to peripheral capillaries.
Other physical parts.
A denser glycocalyx on the blood side, and on the brain side, a basement membrane, parasites, and astrocyte foot processes all contribute to the barrier's integrity.
So what gets across easily?
Small lipophilic molecules, gases, they can diffuse reasonably well. But hydrophilic molecules, nutrients, ions, charged things, many drugs can't diffuse sufficiently.
This is where transporters come in.
Exactly. The BBB needs highly selective transport mechanisms, largely mediated by an enriched set of ABC and SLC transporters in those endothelial cells.
Which ones? Efflux pumps.
Yes, on the luminal blood-facing side, you have ABC efflux transporters like ABCB1, MDR1-PGP, and ABCG2, BCRP. They actively pump out numerous small molecules, drugs, even endogenous stuff like steroids, limiting their brain entry. Table 4.4 summarizes this.
And uptake transporters for nutrients.
Absolutely essential. You need SLC transporters to bring things in. GLUT1, SLC2A1 for glucose, amino acid transporters like LAT1, SLC7A5, and SLC7A1, the folate transporter RFC, SLC19A1.
Others?
Monocarboxylate transporters is SLC16 for lactate pyruvate. SLC22 family members are there too, OATs, SLC22A68, and OCTs, SLC22A13, plus receptor-mediated transport for things like ferret insulin and some low-level transcytosis.
It's a very selective system.
Exquisitely tailored, allows necessary entry, limits xenobiotic drug entry.
Is it also a metabolic barrier?
Enzymes? Yes. Circulating catecholamines are inactivated by endothelial MAO. Endothelial dopa decarboxylase metabolizes L-dopa, why you need an inhibitor for Parkinson's treatment. Gamma-glutamyl transpeptidase cleaves glutathione adducts like LTC4.
How does this affect drug delivery to the brain?
Well, the brain gets about 15% of cardiac output. Small lipid-soluble drugs like anesthetics, nitrous oxide, thiopental, diffuse readily. Some drugs mimic natural substrates and use transporters like LAT1, L-dopa, gabapentin.
What about drugs that are substrates for the efflux pumps?
That's a major problem. Efflux transporters like MDR1, BCRP, MRP4 actively extrude many drugs. Recognition by these pumps is a huge disadvantage for CNS drug development. Charged or large drugs also penetrate poorly.
Can this change in disease?
Research suggests MDR1 BCRP expression in the BBB can be modulated in neurological diseases, potentially contributing to pathology and neurodegeneration.
Are we finding ways around the BBB?
People are working hard on it. Nanotechnology, nanoparticles, liposomes, drug ferritin adducts to use receptor transport, increasing drug lipophilicity. Basic research is also key understanding nuclear receptor regulation of BBB transporter enzymes, how the barrier develops and functions.
Any hints of manipulating the barrier itself?
There's some interesting work. One study showed adenosine A2A receptor activation could reversibly decrease PGP expression in vitro, suggesting maybe A2A agonists could modulate permeability. The goal is controlled, regulated drug entry, which Chapter 17 reviews further.
Okay, let's talk about the extended clearance concept. How does that view liver clearance?
It provides a more integrated perspective. Instead of just thinking about metabolism, it considers the whole dynamic interplay in the liver. Drug uptake into hepatocytes, PS1, backflux out of hepatocytes, PS2, hepatic metabolism, CLMET, and biliary sequestration, or pumping into bile, PS3. Figure 412 illustrates this.
So clearance depends on all these factors.
Right. The overall hepatic intrinsic clearance, Clintol, is described by an equation linking these. Clintol, PS1, CLOMET plus PS3, PS2 plus CLMET plus PS3. That's equation 4.6.
What's a key insight from this?
A crucial one is that if the backflux, PS2, is much smaller than the sum of metabolism and biliary excretion, CLMET plus PS3, which is often true for drugs that are efficiently handled by the liver, then the overall clearance becomes mainly determined by the uptake rate. Clintol is approximately PS1.
So uptake becomes the bottleneck, the rate-limiting step.
Exactly. For many transporter substrates that are well metabolized or excreted, uptake governs how fast the liver clears them.
How does this affect drug exposure, blood versus liver?
This has big implications. For an oral drug cleared by the liver, the equations 4 to 7 for blood AUC, 4 to 8 for liver AUC, suggest that if you decrease the uptake rate, PS1, maybe with an inhibitor, the blood AUC goes up inversely, but the liver AUC might not change much directly.
And if uptake is rate limiting, what happens if metabolism or biliary excretion decreases?
In that case, the liver AUC would likely increase, but the blood AUC might not be affected.
So for drugs like statins affecting liver, side effects may be systemic.
Absolutely. If hepatic uptake clearance decreases due to interaction or genetics, it might mainly affect the risk of systemic side effects like myopathy more than the pharmacological effect in the liver.
Can we model this?
Yes. Physiologically based pharmacokinetic, or PBPK, modeling is invaluable here. It simulates how variations in transporter activity, like OATP1B1 MRP2, affect systemic and liver exposure of drugs like statins.
How do PBPK models work?
They connect tissue compartments using blood flow rates and incorporate detailed info on ADME processes, including specific transporters. They predict the drug's time course throughout the body.
What can PBPK tell us about statins?
Sensitivity analysis using PBPK suggested OATP1B1 variation has minimal impact on pravastatin efficacy in liver, but a large impact on myopathy risk systemic. Conversely, MRP2-BCRP efflux variation might impact efficacy more and myopathy less, as shown in Figure 413.
Does this match real-world observations?
It seems to align. OATP1B1 genetic variation is linked to statin adverse reaction risk, like with simvastatin, while variations affecting biliary excretion or absorption might be more linked to therapeutic response differences.
This leads perfectly into genetic variation in transporters in clinical drug response.
Right. We know inherited defects in specific SLC-ABC transporters cause diseases, tables 4.2, 4.3. But much more common genetic polymorphisms, subtle DNA variations in transporter genes also play huge roles in the variability we see in drug response between individuals. That's pharmacokinetics.
Which transporters have well-known polymorphisms affecting drugs?
Clinical studies consistently show polymorphisms in ABCG2, BCRP, SLCO1B1, OATP1B1, and SLT2A1, OCT1 contribute significantly to inter-individual variation for many drugs. This needs consideration in drug development.
Can you give an example, BCRP?
Yes, a common BCRP missense polymorphism, RS2231142, PGLN141Lis, is linked to increased response to rosuvastatin.
Okay.
BCRP pumps rosuvastatin out in the intestine. This variant reduces function, so more drug gets absorbed. Interestingly, the same variant is linked to poorer response to allopurinol for gout mechanism, less clear there.
And OATP1B1.
The common OATP1B1 missense polymorphism, RS4149056, PVAL174ALA, is strongly associated with higher statin blood levels and increased risk of muscle toxicity, myopathy, as detailed in Chapter 37. It also affects methotrexate and ticagrelor pharmacokinetics.
What about OCT1?
Multiple missense variants in SLC22A1, OCT1, are linked to variable clearance and response for drugs like metformin, sumatriptan, morphine, ondansetron.
Are these the only ones?
No. While OATP1B1, ABCG2, and OCT1 are major players, polymorphism in many other transporters are also associated with drug response variations. For example, tenofovir disposition is linked to polymorphisms in ABCC2, MRP2, and ABCC4, MRP4. It's a widespread phenomenon.
Finally, let's talk about transporters in regulatory sciences. Why are they so important for agencies like the FDA?
Because they're fundamental to drug disposition and action, they're major determinants of therapeutic and adverse reaction variability. And crucially, they mediate drug interactions that can cause serious safety issues.
Like the cerevastatin withdrawal.
Exactly. Gemfibrozil inhibited hepatic uptake and metabolism of cerevastatin, leading to high levels, rhabdomyolysis, and market withdrawal. That highlights the critical need to predict these interactions.
So the FDA has guidance on this.
Yes, they have clinical pharmacology guidance on drug interaction studies during development. It uses in vitro transporter data. Does a new drug inhibit or induce key transporters to decide if clinical interaction studies are needed?
Can you give an example?
If a new drug inhibits OCT2 or MATE-E1 in vitro at relevant concentrations, the guidance suggests considering a clinical study looking at its effect on the renal clearance of a known substrate like metformin in vivo. If no inhibition is seen in vitro, a study might not be recommended.
Which transporters does the current guidance focus on?
OATP1B13, PGP-MDR1, BCRP, OCT2, MATE-E1, OAT1, OAT3. But research is ongoing, identifying other transporters involved in clinical interactions.
Are there ways to measure transporter activity in humans during trials?
Yes, researchers have identified various endogenous biomarkers substances naturally in the body that act as probes for key hepatic and renal transporters. Table 4.5 lists some.
How are these used?
They can be measured in early trials to phenotype subjects, assess baseline transporter function, and evaluate drug-drug interaction potential by seeing if the new drug changes the biomarker levels. This helps plan later studies.
What are some common biomarkers?
Coproporphyrin I is a sensitive marker for OATP1B hepatic uptake. Its plasma levels change if OATP1B is inhibited. N-methylnicotinamide, NMN, is useful for renal OCT2 at peak function. Its renal clearance reflects their activity.
Can modeling help interpret these biomarker changes?
Yes, PBPK and population PK models are being developed for biomarkers like coproporphyrin I. These models aim to translate an observed effect of a new drug on the biomarker into a predicted effect on clinical drugs, like statins, complementing regulatory risk assessment.
So as we wrap up this incredibly detailed deep dive, it's just abundantly clear that membrane transporters are, well, they're far more than just passive gatekeepers.
Absolutely. They're highly dynamic, sophisticated proteins, central and multifaceted roles in almost every aspect of cell function, and especially from our discussion today, in how our bodies interact with medications.
We've covered how they govern everything from initial absorption right through to elimination, how they can be drug targets themselves, and how these subtle genetic variations can lead to this huge spectrum of individual responses.
What's truly striking, I think, is the sheer complexity and scale of this network. Hundreds of transporter genes, each with potentially profound roles, and that coordinated interplay between uptake and efflux in different tissues, all regulated. It just underscores the incredible integration of biological processes.
It really does. Even something as simple as taking a pill involves this immense molecular machinery working behind the scenes.
Exactly.
And hopefully this detailed knowledge isn't just academic for you listening. Understanding these principles gives you a much deeper appreciation for how your own body handles medications, and maybe why responses can vary so much between people.
It can empower you to be a more informed participant in your own healthcare, definitely.
So thinking about all this, what are some final thoughts? Where does this lead?
Well, it raises some really thought-provoking questions. With this incredibly intricate network, much of it still being uncovered, what future breakthroughs might we see in personalized medicine? Can we get to the point of truly tailoring therapies based on an individual's transporter profile, optimizing efficacy, minimizing side effects?
That's the jury.
And with so many orphan transporters still out there, what hidden roles might they play in health and disease? What completely new therapeutic targets might emerge as we finally figure out what they do? It feels like a field that's still brimming with potential for discovery.
Absolutely. A lot learned, but clearly a lot still to explore. If any particular aspect of this deep dive sparked your curiosity, we definitely encourage you to delve further into the scientific literature. It's a rapidly evolving field. Thanks for joining us as we explored the fascinating and intricate world of membrane transporters.